Hello students, welcome to Zoology Classroom. This is L. Mahesh, Assistant Professor of Zoology from Tara Government College, Sangareddy. In today's class, we are going to discuss about regulation of cell cycle. In the previous class, we have seen about different events that occur in cell cycle. In today's class, let us discuss about the events or the mechanisms that regulate cell cycle. First of all, let us know why regulation is required for this cell cycle. We have seen in the last class that cell cycle comprises of interphase and division phase. And again, in interphase, we have G1 phase, S phase, and G2 phase. And in all these phases, different types of activities will occur. For suppose, if the events or the activities that are supposed to occur in G1 phase, let us imagine they have not finished yet. So without finishing those events, what happens if cell enters into S phase? Likewise, what happens before the events that has to happen in S phase? They have to, before they happen, what happens if the cell enters into G2 phase? So unfinished works or unfinished tasks will result in the formation of defective daughter cells. So finally, that is not going to help the organism. So for this purpose, there should be a check. There should be a regulation. So a regulation mechanism is very, very important. If these regulation mechanisms are not there, then it will become very hard for the cell to control its events that happen in the cell cycle. So all these events in the eukaryote cell cycles will be controlled. So three checkpoints are there. Three major checkpoints are there. So they are G1 checkpoint. So at the end of the G1 phase, a first checkpoint will be there. That is G1 checkpoint. And in the S phase, there will be no checkpoint. But at the end of the G2, there will be again another checkpoint, a second checkpoint. This is G2 checkpoint. Once the cell passes this G2 checkpoint, cell enters into division phase. And in the division phase, we know that it is meiosis and mitosis. So in this meiosis and mitosis, prophase, metaphase, anaphase, and telophases will be there. So out of these four phases in karyokinesis, at the end of the anaphase, there will be another checkpoint. This is third checkpoint. It is called M checkpoint. So if the cell passes through this third checkpoint, then only cell will enter into telophase. Otherwise, cell will stay in anaphase only. Cell will be halted at anaphase only. So like this, these three major checkpoints are there. And these checkpoints are also called as restriction points. So how these events all are regulated in the cell? So there is no intelligence in the cell. So all the events are regulated by certain proteins. So the protein synthesis will be regulated. In turn, the proteins will control or they will regulate the events that occur in cell cycle. So the cell cycle is regulated by cyclins. They are special type of proteins. Cyclin dependent kinases, these are also protein, but these are enzymes. They act like enzymes. And cyclin dependent kinase inhibitor, mostly these are also protein in nature, and they will inhibit these cyclin dependent kinases. So they work against to this second one, CDKs. So these cyclin dependent kinase inhibitors are called as CDKIs or CDIs or CKIs. So these three abbreviations are for this single thing, that is cyclin dependent kinase inhibitors. And fourth one is RB proteins, retinoblastoma proteins, and the fifth one is P53 gene. So out of these five things, first two things, cyclins and cyclin dependent kinases, CDKs, are called as positive regulators, And remaining 3, cyclin-dependent kinase inhibitors, RB proteins, and P53 gene. These 3 are called as negative regulators. So coming to the cyclins, what are cyclins? Cyclins are one type of proteins. They will be usually globular in nature, and 4 major cyclins are present: cyclin A, cyclin B, cyclin E, and cyclin D. So C is not present. C will be there, but it has no role in controlling or regulating the cell cycle. So these A, B, E, D, these 4 are regulating cyclins. And the concentrations of these 4 cyclins varies during the cell cycle. So the concentration is not constant. The concentration will be changing depending on the phases of the cell cycle. And what they will do, these cyclins, they will phosphorylate CDKs. CDKs, as we have already told, they are special enzymes. They will control the events that occur in cell cycle. So these CDKs will be activated only when they are phosphorylated. Phosphorylation means transferring phosphate group from ATP to these CDKs. So this transferring of phosphate group from ATPs to the CDKs will be done by cyclin proteins. So cyclin proteins are very, very important. When cyclin proteins are fully formed, then CDKs will be phosphorylated. When CDKs are phosphorylated, they become activated. When they are become activated, the required protein synthesis will be done for the events that has to happen next. So in some orphan cyclins also will be there. They do not have any CDK partners. So cyclin A has a special partner, CDK partner, cyclin B, cyclin E, cyclin D. Each of these cyclins, they have a special cyclin-dependent kinase partners. But some cyclins, they do not have any CDK partners. Such type of cyclins are called as orphan cyclins. For example, cyclin F, this is orphan cyclin. It is essential for G2 to M transition, but it is not having any CDK partner. And these cyclins, again, they are considered as 2 types. They are classified into 2 types. One is G1 S cyclins, another one is G2 M cyclins. So G1 or S cyclins, examples for this G1 S cyclins are cyclin A, D, and E. And for G2 and M cyclin B is example for that. And second thing, cyclin-dependent kinases, CDKs. So these are also proteins, and these are enzymes. They regulate the events in this cell cycle. So these are required for some activities to occur. So, but they will be in inactive form. These will be activated only when they are coupled to the respective cyclin. Here we can see what are the cyclins and what are the counterparts are paired CDKs. So in G0 phase, cyclin C will be there. So when cyclin C is produced, cell will enter into G0 phase, as we have told in the last class. And counterpart CDK is CDK3. And in G1 phase, cyclin E will be there mostly. And at the end of G1 phase, cyclin E also will be produced. And the cyclin D will be controlled by CDK4 and CDK6. Cyclin E also will be controlled by CDK2. So this CDK4, 2, and 6 will control, or they will be activated by cyclin E and cyclin E. And in the S phase, cyclin A will be produced and cyclin E will be produced. And these 2 cyclins, they will activate cyclin-dependent kinase 2. In G2, cyclin A will be there, and it will activate cyclin-dependent kinase 2 and cyclin-dependent kinase 1. In the M phase, B cyclin will be produced, and this B cyclin activates cyclin-dependent kinase 1. And third regulatory molecules are cyclin-dependent kinase inhibitors, CDKs or CKIs or CDIs. So they will inhibit the cyclin-dependent kinase enzymes. So they act against to the second enzyme, second molecules, cyclin-dependent kinases. So they bind to the cyclin-dependent kinase enzymes and cyclin complexes. So cyclin and cyclin-dependent enzyme complexes will be bind to these CDKIs. So when CDKIs are bind to cyclin and CDK complexes, they will block the activity of what CDKIs, CDKs. So these CDK enzyme activity will be inhibited when these CDKIs are bound to this complex. So in this way, the CDK4 and CDK6 will be inhibited by this CDKIs. The CDK4 and CDK6 are very, very important for the cell to enter from G1 to S phase. So when these two CDKs, CDK4, CDK6 are inhibited, cell will stay in G1 phase only. It will not enter into G0 phase. In this way, they will control the cell regulation or cell cycle events. So, and the CDKs are of two types. They are, one is INC family or inhibitors of kinases. P15, P16, P18, and P19 are example for this. And second family is CIP family, CDK inhibitory proteins. P21, P27, P57 are examples for this second family. And these inhibitors totally P16, P15, P18, P19, P21, P27, P57. These are the cyclin dependent kinase proteins, kinase inhibitory proteins. P16 inhibits CDK4 and CDK6. P15 inhibits CDK4. P18 inhibits CDK4 and CDK6. P19 inhibits CDK4 and CDK6. P21 inhibits cyclin E1 and CDK2. P27 inhibits CDK3, CDK4, CDK2, and cyclin E1. And P57 inhibits cyclin E1 and CDK2 enzyme. So another, the fourth component that controls the cell cycle, that regulates cell cycle, is RB protein. RB protein, first it was identified from a gene, that is RB gene. This RB gene controls one type of tumor. So it suppresses tumor. So it is called tumor suppressor gene. So the protein synthesized from this gene is called RB protein, retinoblastoma protein. So this retinoblastoma protein, very, very important one. It binds with G1 cyclin and respective CDK complex. So when this RB protein is unphosphorylated, it is not bind to phosphate group. Then this RB will be attached to E2F. Elongation factors, as I have already told in the last class, elongation factor is very, very important for the protein synthesis. So these elongation factors, okay, what elongation factors? Elongation factors required for the synthesis of cyclin D. Okay, elongation factors required for the synthesis of cyclin D and cyclin E. So these cyclin proteins require the elongation factors. So if the cyclin proteins are to form from protein synthesis, required elongation factors should be activated. But these activated factors are bind to RB. When they are bind to RB, elongation of the cyclin D protein, cyclin E protein will not take place. So that what happens, due to the lack of the cyclin D proteins, the cell fails to pass the G1 checkpoint. When the cell passes G1 checkpoint, then only cell enters into S phase. So to pass the checkpoint, to pass the G1 checkpoint, what has to happen? Cyclin D component has to fully formed, and cyclin E also should be initiated. Okay, before the cell enters into S phase, cyclin E synthesis will be initiated. So cyclin E production should be started. Cyclin D production should come to an end. So cyclin D production should be finished, and cyclin E production should be initiated. So the production of cyclin D and cyclin E will be inhibited as RB protein is bind to E2Fs. When E2Fs are bind to RB proteins, cyclin D synthesis, cyclin E synthesis will not be finished. So that what happens, cell will stay in G1 phase only. So in this way, RB protein is controlling the cell to not entering. It will not allow the cell to enter into S phase. So instead, it allows the cell to stay in G1 phase. So and another one is P53 protein. P53 protein will be synthesized from P53 gene. So this P53 gene is very, very important one. It is responsible for apoptosis in the damaged cells. So this keeps a check on the DNA content or DNA damage. If the cell DNA is damaged, and if the DNA damage is irreparable, then what happens? P53 promotes some events. These events, they will result in the death of the cell, that is called apoptosis. So apoptosis is nothing but programmed cell death. So cell wantedly commits suicide with the help of this P53 gene. So all cells will not commit suicide, or all cells will not become damaged or hydrolyzed or digested. So only such cells, they have irreparable DNA damage. Such type of cells only, they will undergo into this apoptosis process. So how this thing is controlled is that P53 gene will be controlled by a protein called MDM2. MDM2 protein attaches to this P53 gene. When DNA damage has happened, then what happens? DNA damage or cell cycle abnormalities. Okay, in the events that has to happen in cell cycle, if any abnormalities are there in these events, then automatically this MDM2 will be released from P53 gene, and P53 gene produces P53 protein. So under hypoxia conditions also, P53 gene will be activated, P53 protein will be produced. So this P53 protein, what it will do, it will arrest the cell cycle. It will not allow the cell to enter into S phase, or it will not allow the cell enter into division phase. And in the division phase also, during the division phase also, before the third checkpoint, if the cell fails to pass that checkpoint, cell cycle will be arrested in that same stage, and DNA repair mechanism will take place. If the DNA repair is successfully finished, then what happens? Cell cycle will be restarted. If the DNA repair mechanisms are abnormally, if the DNA repair mechanism is failed and abnormalities are not corrected, then what happens? The cell will decide that the cell is useless and it should not undergo division. So when that decision is taken place, P53 gene promotes apoptosis. So such type of damaged cells, abnormal cells will undergo apoptosis and they will be dead and they will be eliminated. So in this way, cellular and genetic stability will be assured by the P53 protein. So in this way, P53 protein also controlling the cell cycle. And here in this slide, you can see that on y-axis, we have taken concentration of the respective proteins, and on x-axis we have taken different phases. So G1 phase, S phase, G2 phase, and mitosis phase or division phase. So in all these phases, what are the concentrations and what are the levels of these different cyclin proteins? So red one is cyclin D, green one is cyclin E, and blue one is cyclin A, and the yellow one is cyclin B. So here we can see, as per this graph, that uh, at the beginning of the G1 phase, cyclin D will not be at all. That means cyclin D concentration will be 0, and the production will be initiated. At the end of the G1 phase, cyclin D production reaches maximum stage, and the cyclin D, okay, the cyclin D molecules already produced in G1 phase, they will continue to survive throughout S phase and G2 phase. At the end of the G2 phase, the cyclin D proteins will start damaged, or they will start disappearing from the cell. So before the cell finishes this mitosis phase, cyclin D concentration completely comes to 0 point, or it will be completely disappear from the cell. And coming to cyclin E, so in the middle of the G1 phase, cyclin E production will be initiated. At the end of the G1 phase, cyclin E production reaches maximum. So all the cyclin proteins will be synthesized by the end of the G1 phase and at the beginning of the S phase. And in the middle of the S phase, this cyclin E concentration will start to decline, and by the end of this S phase, we cannot see any cyclin E in the cell again. So cyclin E will be synthesized in the middle of the G1 phase, and it will be controlled or damaged or disappeared from cell in the middle of the S phase. And coming to cyclin A, cyclin A also will be started synthesis of cyclin A will be initiated in the late G1 phase, and it reaches maximum production level in the middle of the G2 phase, and in the beginning of the mitosis phase, cyclin A will disappear from the cell. And coming to cyclin B, synthesis of cyclin B will be initiated in the beginning of S phase, but the production will reach maximum point or saturation point in the beginning of the mitosis phase. And in the middle of the mitosis phase, cyclin B completely disappears from the cell. So these are the concentrations. So these concentrations are very, very important for cell cycle to occur normally. So if these graphs are not in this way, that means abnormalities are there in the cell cycle. So this is the normal cell cycle graphs for these concentrations of for these cyclins. So here in this graph also, in this chart also, you can see that in the mitosis phase, we have not seeing in this thing only this third checkpoint, mitosis checkpoint. So in the first checkpoint, we can clearly see that. So G1 phase, in the G1 phase, at the beginning of the G1 phase, cyclin D synthesis will be initiated. At the end of the G1 phase, cyclin D reaches maximum production stage, and they will bind with cyclin dependent kinases 4 and 6. So in the middle of the G1 phase itself, cyclin A production will be initiated, and it continues up to G2 phase, and this cyclin E reacts with CDK2 enzyme. And in the middle of this S phase, cyclin A production will be initiated, and it will continue till the beginning of mitosis phase or division phase, and cyclin A binds with cyclin dependent kinase 1 and 2. In the beginning of the G2 phase, cyclin B synthesis will be initiated. It binds with CDK2, and in the beginning of mitosis phase, this cyclin B will be disappeared from the cell. So first one is G1 checkpoint. So G1 checkpoint is present at the end of the G1 phase and before the S phase. It helps in taking decision whether the cell has passed all the requirements. That means like whether the cell should enter into division or the division should be delayed, or the cell should enter into resting phase. That means G0 phase. So all these decisions will be taken at G1 checkpoint. So if unfavorable conditions are there for the cell division, so if the cell division is not possible, then the G1 phase will be restricted and it will be arrested. If the cell is not able to enter into this division phase for a longer period of time, then the cell will be diverted into G0 phase, where no progress will take place in this G0 phase. So here what happens, what is the checkpoint, what is the restriction point? Cyclin D and cyclin dependent kinase. Okay, cyclin D and cyclin dependent kinase. So the concentration of the cyclin D and cyclin dependent kinases must reach the maximum concentration. Then only cell will be allowed to enter into the next phase, that is S phase. So here the controlling thing is cyclin dependent kinase inhibitor P16. So cyclin dependent kinases, various types are there: P15, P16, P18, P19, and P21, P27, P57. These are the cyclin dependent kinase inhibitors. Out of them, P16 inhibitor will be attached to the cyclin D and cyclin dependent kinase 4 and 6 enzymes. So they will attach to these cyclins and enzymes. When this P16 is attached to these cyclins and CDK enzymes, then cell will not enter into S phase. So P16 protein concentration, if it is high, and if this P16 protein is attached to the cyclin D and CDKs, then cell will not allowed to enter into S phase. And active cyclin D, cyclin CDK complexes, they will phosphorylate retinoblastoma protein. If retinoblastoma protein is bind to this cyclin D and CDK complexes, as we have already seen, this RB protein or PRB, retinoblastoma protein, it inhibits the G1 by preventing E2F elongation factors required in the transcription of the cyclin E and cyclin A. So cyclin E and cyclin A, they will be required in the S phase. Okay, in the synthesis phase, cyclin E, cyclin A proteins are required. So for the production of cyclin E and cyclin A, and for the production to reach maximum level, for the production of cyclin D to reach the maximum level, also elongation factors are required. So all these elongation factors required for cyclin D production, cyclin E production, cyclin A production will be inhibited by PRB. When the PRB becomes phosphorylated, this PRB separates from these elongation factors. When these elongation factors are separated, cyclin D, cyclin E, cyclin A will be synthesized. This synthesized cyclin E's, A's, and cyclin D's. All these things, they will bind with this respective CDK enzymes, and automatically cell enters into G1, from G1 to S phase. So in this way, this RB protein, okay, RB protein also plays a very important role in checking this G1 checkpoint. So, and the next one is G2 checkpoint. Second one is G2 checkpoint. This G2 checkpoint is present at the end of the G2 phase. It checks the number of factors which are essential for cell division. So out of them, maturation promoting factor MPF, or mitosis promoting factor, or M phase promoting factor. MPF is very, very important one. MPF is nothing but cyclin B and CDK1. So cyclin B and CDK1 complex is called as MPF, maturation promoting factor, or mitosis promoting factor, or M phase promoting factor. So cyclin B production should reach maximum level, then only this MPF will be formed. When MPF are fully formed, then only the cell will enter into division phase. That means the cell will pass the G2 checkpoint. So in this way, G2 checkpoint sees the concentration of MPF. When the MPF concentration is full, then G2 checkpoint passes the cell to enter into division phase. So in this way, not only MPF, another enzyme also is present that is called phosphatase, CHK phosphatase. So this phosphatase helps the cell to enter into division phase or M phase by removing the inhibitory phosphate group that was added to this phosphatase. So this phosphatase will be attached to this MPF. When phosphate group is removed from this phosphatase enzyme, then MPF will be activated, and this activated MPF allows the cell enter into division phase, that is M phase. So, and next check in this G2 checkpoint, what is the function of this MPF? Is that MPF maturation promoting factors are cyclin B and CDK1 complex helps in triggering the formation of spindle fiber. Spindle fiber formation is very, very important in the cell division. And this MPF also promotes chromosome condensation. So chromosomes, they should be condensed, and they should reach the, or they should get the, or they should acquire the chromosome form. That also will be promoted by this MPF. And nuclear membrane will be disappeared with the help of this MPF only. So these are the functions of MPF. If any damages are noticed, then what happens? Phosphatase enzyme will not be phosphorylated. Then it will not lose the phosphate group. Then MPF will not activated. Inactivated MPF results in the arresting of the cell cycle in G2 phase only. So division phase will not be entered. The cell will not enter into the division phase. What are the damages? And all those damages should be repaired. Only after the repairs work is done, then only this phosphatase group will lose phosphate. Phosphate will add to this MPF. Then MPF will be activated. Then activated MPF will allows the cell to enter into division phase. So next one is a metaphase checkpoint, or M phase. So this will be present in the at the end of the, or during the anaphase of the division phase. So in the anaphase promoting complex will be formed. This anaphase promoting complex regulates the spindle checkpoint. So this is also called a spindle checkpoint. So formation of anaphase promoting complex only allows the cell to enter into telophase. So here what happens, this anaphase promoting complex checks whether the chromosomes attached to the spindle fiber correctly or not. In the previous classes, we have seen that during the mitosis, each chromosome will be attached to two opposite spindle fibers. But in the meiosis one, each chromosome will be attached to only one spindle fiber. In the pair, one chromosome will be attached to one centriole. The other chromosome of the same pair will attach to the opposite centriole. So this is the proper attachment of chromosomes in respective mitosis and meiosis phases. So if this attachment of spindle fibers is accurate or not, it will be checked by this anaphase promoting complex. And this anaphase promoting complex also, it governs the alignment of chromosomes. So chromosome should be arranged in the required way. So we have seen that in the mitosis, all chromosomes will form a single layer exactly in the middle of the cell. But during the metaphase 1 of meiosis 1, the chromosomes will form two layers. So the alignment of these chromosomes, whether it is correct or not, so this thing also will be controlled by anaphase promoting complex. And if there are any mistakes, okay, if there are any mistakes in this formation of spindle fibers, alignment of chromosomes, then what happens? The cell will be arrested in this anaphase only. Only when the mistakes are corrected or deficiencies are fulfilled, then only the cell will be allowed to enter into telophase. Then only the division will be completed. Otherwise, division will not be completed. So in this way, this metaphase checkpoint or metaphase restriction point controls the cell's entry into telophase from anaphase. And DNA damage also will be controlled. DNA damage also will be checked with these checkpoints. So DNA damage checking will be done at G1 checkpoint and G2 checkpoint. Here at these two places, this thing DNA damage will be checked. So in the metaphase, only the chromosome alignments and spindle fiber formation will be checked in the third checkpoint, that is metaphase checkpoint. In the first two checkpoints only, DNA damage will be checked. So if the DNA is damaged, then the cell will be arrested in G1 phase or in G2 phase. So and then what happens? The cell attempts to correct the DNA damages. It will try to repair those DNA damages. Only when the DNA damage repair work is done, it is finished, it is successful, then only the cell will enter into the next phase. Otherwise, the cell will be allowed to undergo this apoptosis. That means cell destruction, cell suicide will be done with the help of P53 gene. So P53 gene controls the checking points here at G1 checkpoint and G2 checkpoint. So the major work of P53 gene is to check the DNA damage. So damaged DNA will not result in the entry into the subsequent stages. So this thing will be done with the help of P53 gene. And DNA damage can be done in the uh, in the what yes phase. Here it's the G2 checkpoint. DNA damage could happen due to UV radiation, oxidative stress, etc. All these damages will be checked in the S phase, and in the G1 checkpoint, DNA damage may happen with due to various regions. So that DNA damages will be checked at the G1 checkpoint, and entry will be restricted into S phase here in this given checkpoint. So, and once this second checkpoint is allowed the cell to enter into this thing, then there will be no DNA repair mechanism will be happening in the cell. Only anaphase promoting complex, it will control the events of this spindle fiber formation and chromosomal element on all those things. And in the S phase, what happens? DNA replication stops at any point of the DNA. the progress through the cell cycle also will be halted until the problems are solved. Only after the problems are solved, then only the cell will be allowed to enter into the subsequent stages or phases. So in this way, all these things are controlled in the cell cycle. So the concentration of these cyclin proteins, the concentration of inhibitor proteins, and this formation of anaphase promoting complexes, all these things are controlling the cell cycle events. So with this, today's topic is over. Let us meet in next class with another topic.